Good afternoon, good evening, and good morning, everybody, wherever you might be. I am Rob Burgess, and I head up business development for Sinobiological. And it is my pleasure and honor to be the host today of BioTalk Tuesdays for Sino. We have an extremely interesting and exciting topic that's going to be discussed today by one of the world's leaders in microRNA delivery. It's specifically photo control of microRNA delivery as it pertains to cancer therapeutics. So before we get to that exciting seminar, I just wanted to mention one brief housekeeping issue, and that is related to questions. If you have a question, we ask you to simply type it into the chat box. And if we have time at the end of the seminar today, I will ask those questions verbally, as many as I can, of our speaker and give the speaker a chance to respond. I do know the speaker has a tight schedule today, so any questions we can't get to, we will make a log of the chat box and we will ask the speaker to answer them individually outside of the meeting. So without further ado, it is my pleasure to introduce to you all our speaker today. It is Dr. Dan Hayes. Dan is a faculty member in the Biomedical Engineering Department at Penn State University, and he is also the department head of the Huck Chair of Nanotherapeutics. In addition to this, he is the director of the Center of Excellence in Industrial Biotechnology, and he has also co-founded numerous university spinouts, which include Nano Horizons Incorporated, as well as Illuminate Therapeutics. Dan has authored over 120 peer-reviewed publications and is an inventor on 15 pending as well as allowed patents. It is quite an honor to have Dr. Hayes here today, and the title of Dr. Hayes' talk is Photocontrolled Delivery of miRNA-34a, which mimics the drive of apoptosis in esophageal cancer cells and regression of tumors in vivo. And so with that, Dr. Hayes, I will turn the screen sharing over to you.
Thank you, Rob. It's my pleasure to be here. All right, let me see. One second. All right, can everyone see that?
Yeah, it looks great. Thanks.
Excellent. All right, so as Rob said, I'm going to talk a little bit about some of the photoactivated siRNA delivery technology that we've developed. And particularly, I'm going to talk about this technology as a platform for delivery of microRNA mimics in driving the apoptosis of cancer and reduction of tumors in vivo. So just a little quick background on antisense gene regulation. So these are short non-coding sequences, antisense oligonucleotides, siRNAs, microRNA mimics, small hairpin RNAs. These genes are complementary to one or more messenger RNAs in biological systems. They downregulate the expression of those. In general, they downregulate the expression of those via translational repression, cleavage, or deadenylation. They can also upregulate the expression of certain genes, depending on where they impact in the promoter region. But in general, it's a degradation process. And typically, they're going to interact, particularly the microRNA mimics that we are making, that are mimics, siRNA that are mimics of the naturally occurring microRNA, interact with the RISC complex and bind to the 3' UTR of messenger RNA, driving that degradation process. One of the interesting things about microRNA mimics is that they get a substantial boost in terms of their efficacy because one single microRNA in the RISC complex can degrade hundreds, if not thousands, of messenger RNA. So you get a lot of bang for your buck in the delivery of messenger RNA if it's done appropriately.
Hey, Dan, please forgive me for interrupting you. I'm very sorry. Would you mind putting your presentation in full, like presentation mode for everybody?
Sure. Can you see that?
Yeah, I think that works well. Let me just ask Christine and Lilith, does that look good to you?
If you can make it so it's full screen, we can see it in presenter mode as well, if you don't mind making it like full screen presentation.
Oh, don't know how to do that. How do I do that?
You go up to the top and it's reverse screen, I believe. There you
There you
go.
go. Yeah, looks great. Thank you so much.
Apologize for that interruption, Dan. Thank you.
No worries. You'd think after several years of teaching on Zoom during the pandemic that I would be better at this, but well, I apologize all. And so the system that we're using is we developed is this light inducible gene regulation system. LIGER is the acronym that we use. And so the challenge in delivering siRNA or microRNA mimics has been widely reported. The PK/PD of these nucleic acid therapeutics is not great. So there's a lot of off-target effects, a lot of side effects, and just general lack of stability in vivo. These things degrade very rapidly. The bulk of them end up in the liver, one way or another. And so to use some of these relatively potent microRNA, we tried to develop this spatially and temporally controlled system, and we used light as our trigger. So this responds to a very specific wavelength of light based on the plasmonics of the particle that we've developed. And what's really important about this is it develops this step function-like release profile. And this is based on the external or the cleavage moiety that we use, which is a Diels Alder compound. And it has a very specific barrier for its retroreaction. And below that barrier, we see very little activity. And above that barrier, we see this dramatic release of almost all of the compounds. So you get this step-like function. How do we make this? So we start with a nanoparticle, metal nanoparticle or plasmonic particle. We attach our Diels Alder compound to that. There are a number of different flavors of Diels Alder compounds that we've explored, which I'll talk to you about. Then we add, through just a simple EDC reaction, we add on the siRNA or RNA sequence to the end of it. What's interesting about Diels Alder compounds and why we're so interested in them is they have very specific reverse reaction barriers, as we said, but also when they go through their retroreaction, you get back the starting materials. So we don't have a random distribution of different cleavage materials with different toxicities. We're getting back the starting materials, which makes predicting the cytotoxicity and biocompatibility relatively straightforward. And this is done through heat. So as you add energy to the system, and we'll talk about the different actually forms of energy that we're adding to the system, because it's not just heat. But as you add energy to the system, when you reach that threshold, you go through that retroreaction. The leaving group that we use in all of our compounds is a maleimide. So this is the piece that goes with the siRNA. And we found that that is well tolerated in our system and doesn't interfere with the engagement in the risk complex and degradation of the messenger RNA. So we explored 3 of them in this study. As I said, our dienophile is always the same. Our diene, we either look at a furan-based diene, a thiene-based diene, or a pyrrole-based diene to make our Diels Alder compound. They have very predictable reverse reactions, as I said, and they span a large physiological range. So our furan-based Diels Alder is the compound that falls in the middle of our energy range. Our thiene-based compound has the highest barrier for its retroreaction, and our pyrrole has the lowest reaction. So if you think about them, the pyrrole is most likely to degrade, the thiene is the least likely to degrade. And as we put in energy, you see that's the case. And so this is just a simple water bath study where we looked at the stability at different temperatures. And as you can see, it follows that trend. So our pyrrole degrades most rapidly, our furan degrades second, and then our thiene is the least likely to degrade. We also used SHG to gain kinetic information on the cleavage of the different linkers. So SHG is a second harmonic generation. This is when 2 photons combine at the surface of a particle to create one higher energy photon. The occurrence of this is nonlinear and is very highly dependent upon the surface composition of the particle itself, specifically the ionic composition. So as the ionic composition changes, the likelihood of getting an SHG event also changes. This allows us to track in real time the release of the RNA because the RNA is highly charged. And you can see over time that as we heat just the pyrrole, it degrades over time, which would be expected. And so we can track this in real time. And so if we look at it, we can see what the release kinetics of these are. And again, it follows the trend that we would expect. The pyrrole, the furan, and thiophene follow that predictable path. The pyrrole cleaving very, very rapidly, the furan next, and the thiophene next. And again, we can see this in actual real time. So we start to see this cleavage over a matter of seconds. In a matter of minutes, the entire reaction is done. For this study, we developed a new core shell plasmonic particle. In this case, it is a gold shell, a gold core with a silver shell followed by another silver shell or gold shell. And the purpose of this is to create something that is both high, it has a high photon capture cross section, is mechanically very efficient to create phonons, and is still in the size range that is going to be taken up readily by tumor cells. So we start with this gold core. It's about 4 nanometers. We add a silver shell to that, takes it out to about 50 nanometers, then add on another gold shell, which carries it out to about 70 or 80 nanometers, and shifts the wavelength of absorption from approximately 535 for the gold core to around 430 with the gold silver shell. When we add on the gold shell, it moves that all the way out into the near IR. Then we demonstrated the photothermal activation of this compound. And so similarly, using SHG, we could track it and look at the energy dependence as we added in 860 nanometer light. And as you can see, as we increase the amount of light that we're putting into the system, we increase the cleavage rate. We also looked at its general stability in just different temperature environments. And again, this is the furan-based Diels Alder. And you can see it doesn't change radically between 25 and 40 degrees, but as you go above 40 degrees, we start to see that cleavage event. And again, what we're trying to demonstrate here is that it's going to be stable under general physiological conditions, but when we put energy into the system, we're going to get a predictable cleavage. If you look in the center bottom, you'll see a comparison of our core shell nanoparticles and gold nanoparticles at the resonant frequency. And the core shell particle is just vastly more efficient in terms of releasing our Diels Alder. And this is because of the structure itself. The plasmons that are bouncing around inside the particle tend to recombine at interfaces. And since we have these multiple interfaces within our structure between the gold, the silver, and the gold, we tend to get efficient recombination and generation of phonons or heat. In the bottom right-hand corner, you can see a release study from our siRNA in this system. And you can see we start to see substantial release at just 5 joules of energy. This is important because that means we don't have to deliver a lot of energy into the body at the appropriate wavelength to drive this cleavage reaction and deliver our siRNA intracellularly. The less light we have to deliver, the greater penetration depth that we're going to get for any given power of light. So we conducted our first set of experiments in a immune-suppressed tumor microenvironment. We were using the K14 promoter region to drive a TET HRAS-driven tumor in a mouse. And this is on a C57 black mouse base. We injected them with 3 milligrams per kilogram of microRNA-148b liger. This is, again, our light-activated gene regulation system, which equates to about 50 nanomolar microRNA-148b mimic. And the reason we chose microRNA-148b in this study is just microRNA was demonstrated to be downregulated in tumor cells. And this was being used as a diagnostic and prognostic for cancer purposes, but it's also known to be a tumor cell suppressor. microRNA-148b plays a number of different roles in cancer. There's a number of different pathways in the KEGG cancer pathway descriptions. And this is why we like to use microRNA mimics as opposed to just simple siRNAs for specific messenger RNA. We get a lot of bang for our buck out of microRNA because they are conserved evolutionarily, and they have a lot of impact on a number of overlapping pathways. In fact, if you look at microRNA-148b and you look at its predicted gene targets, there's thousands of total genes that it targets. And over 300 of them are in the oncogene class. Why we also need spatial and temporal control of this process, because there's thousands of targets for microRNA-148b. And you can imagine the side effects of a mimic could be very, very dramatic depending on the tissue type. So we looked at uptake into cells, and we found that we used endosomal tracking to determine where our particles were ending up. And so we're using the red as the endosomal tracker. The green is the actual nanoparticle, false colored in this. And this is just the backscattering from the particle itself. It appears because of that plasmonic resonance in our fluorescent channels at the appropriate wavelength. What we don't see is a lot of overlap. So this is not a simple endosomal pathway. We're seeing a number of different pathways that are involved in taking up nanoparticles. And this agrees with previous literature that had looked at the uptake of nanoparticles in cell-based systems. We do drive apoptosis. And so in this case, we're looking at primary keratinocytes. We're looking at our RAS expressing keratinocytes and at PAM212 as a squamous carcinoma mouse line for comparison. And you can see we don't see a lot of impact in our primary keratinocytes. We do see downregulation our RAS expressing keratinocytes and PAM212 when we deliver our microRNA-148b via our nanoparticle delivery system. The non-coding one here, so SNP-DANC is just a scrambled version of that microRNA-148b, which you would expect not to have an effect on these cancer pathways. It does have a number of different other targets because it's only 21 base pairs. And even if you scramble it, you have some targets, but they are not in the cancer pathways that we're looking at. It doesn't have an impact on it. Then we also compared it to lipofectamine delivery and the negative control vehicle delivery. You can see on the right, which is just a quantification using pico green of the number of cells that are left. And you can see that substantial downregulation compared in the far right-hand column, primary keratinocytes compared to our RAS expressing keratinocytes and PAM212. So our primary keratinocytes are unaffected. Our RAS expressing keratinocytes and PAM212 cells are dramatically affected by the delivery of this microRNA mimic. We also looked at the direct targets of microRNA-148b. So ALCAM, ITGA5, ROC1, NRP1, WNT1. These are not all, as I said, there are hundreds of targets, if not thousands of targets, but hundreds within the cancer pathways. But we picked 5 that we thought, based on their target prediction, would be very, very likely real targets. And all 5 of those were knocked down substantially in our experimental group versus our control groups. Also, this is a apoptotic process, as demonstrated by cleaved caspase upregulation. So now the really important data, the tumor regression itself. So if we look at the distribution of our nanoparticles, and here we're just tracking the metal content of the different tissues, you can see that the vast majority of them go to the spleen, very little go to the tumor. And this was an IV retroorbital delivery route of administration. We do see some accumulation in the liver over time, and we see a lot of them actually get taken to the spleen, and we believe this is by immune response. We have a nanoparticle, a 70 or 80 nanometer particle that has a substantial amount of nucleic acid on it. We believe it's being recognized by immune cells. We'll actually show some data in a moment that demonstrates that there is a change in the phenotype of immune cells. But importantly, what we see in the time of administration here is noted as day 1. If you follow the blue line in the middle panel on the top, you'll see that by day 2, so 24 hours later, we've lost about 95% of the tumor volume. By day 3, we've lost 99% of the tumor volume. So the tumor has collapsed dramatically after administration and light activation. That light activation occurs 6 hours after administration. And that was based on our basic PK data. If we look at the nanoparticle non-coding group or scrambled group before and after treatment, we don't see any difference in the light activated versus non-light activated. And the animals seem to maintain their relative healthy weight with respect to time. When we look at the tumors themselves, the tumors are substantially degraded. So if we compare our PBS at the top left as our vehicle control, and then we look at the nanoparticle with the non-coding, we don't see a substantial amount of difference in tissue structure. But if you look at it with the microRNA 148b delivery and light activation, you see that this tissue has become Swiss cheese. It is highly degraded, which is not surprising based on the images that we just saw. The entire tissue volume is gone within about 48 hours. We also looked at some of the other tissues to see if we could see any obvious toxicity, and we really couldn't. So we didn't see any meaningful differences between our vehicle controls, our light activation, scramble controls, or the experimental group. As I said, we do see some immune activation, and we see a substantial amount of infiltration by immune cells. And this is not surprising. Again, given the speed at which the tumor is collapsing, you would expect there to be a substantial amount of immune infiltration. So if you look at our general population of immune cells, CD45 positive, obviously you see a tremendous amount, but we also see specifically CD8 positive, CD4 and CD3 positive cells entering that space, and a substantial upregulation of cleaved caspase. And if you look over on the right, you can see that is a cut through the tumor, the intersection of the tumor tissue with the normal skin tissue next to it. And you can see that the immune cells are actually migrating from the healthy tissue into that tumor tissue as it collapses. As I said, we saw a change in the immune cell phenotype. And so we see a upregulation of Th1 cells in the tumor itself. We don't see a substantial, these are very difficult experiments to do with the tumors changing so rapidly, but we were able to at least capture an upswing in the tumor itself, but also in the spleen, which was much easier to get the data from. We see a change in the phenotype and a significant upregulation of Th1 and Treg cells. So there's a change in the phenotype upon both delivery. So the non-coding, just having the nanoparticle in the system causes a change in phenotype, and then the light activation itself causes this change in T regulatory. We did some basic ADME tox. We don't see a lot of toxicity. So we actually couldn't find any differences between our reference, our vehicle control, any meaningful differences or significant differences, and our two control groups in our experimental group. So all the liver markers look good, Creed, Bun, all asked. But then when we also looked at our hematology, we don't see really any changes across the board. So everything is within very close to the reference values and very close to our no meaningful differences between our control groups and our experimental group. So that was all done with a mouse model system in a mouse tumor, even though it was an HRAS gene being driven by the K14 promoter. Here, we're actually doing a human xenograft. So we looked at squamous carcinoma of the esophagus, and that is the TE10 line, and we did a xenograft in a nude mouse model. And you can just see the illumination method up there on the right. This is near IR, and you can see it in this image, but you can't see it in real life. Our phones, this is taken by a phone, and a phone is capable of seeing into the near IR. So it's human esophageal squamous tumor, immune-incompetent mouse. And again, we're injecting with 3 milligrams per kilogram, but in this case, miRNA-34a, but at the same concentration, so 50 nanomolar miRNA-34 mimic. Why do we choose miRNA-34a? We did some target predictions. So we looked at both the microRNAs that were downregulated in esophageal tumors, squamous esophageal tumors, and the messenger RNA of cancer genes that were upregulated. We took that 2 data sets and looked at the intersection of them. And we came up and then also looked at what genes were predicted to be within the cancer pathways, or what microRNA were predicted to be in the cancer pathways. And we came up with a short list of 5 microRNA. We eventually settled on miRNA-34a5p, and I'll show you that data in a moment, why we picked that one. We also looked at the intracellular release, so cellular uptake and intracellular release. We get about a 92% transfection efficiency or uptake efficiency, depending on how you want to describe it. And you can see that in A and B. And then in C and D, we have C is our Prior to light activation, that red color that you see is the nanoparticle itself and the reflection of scattering that comes from the plasmonic particle. And then in D, after light activation, the change in color, so this is a combined green and red channel or near IR channel, you can see that we have an upregulation of green fluorescence or an increase in green fluorescence, which is the fluorescent tag that is on our siRNA. When it's released from the particle after light activation, the particle is unquenched, or the fluorophore is unquenched, and then you can see it. And so this just demonstrates that we are able to do that release intracellularly. We looked at all of the microRNA that we had identified in our bioinformatics assays, and we tested them all against TE10. And miRNA-34a had the greatest response at the lowest concentration. And you can just see this here. This was just scanning our different microRNA candidates against the TE10. And this was all done with the nanoparticle system itself. We then optimized that dosage for our in vitro experiments and found again that 50 nanometers was approximately correct. And this is the results that came from that 50 nanometer study. And you can see the live dead study here at 72 hours. This is the experimental group, which is again, we're using the same scramble of miRNA-34a as our non-coding. The same scrambled material does not show response. Our CSS miRNA-34a plus light does show substantial response, very similar to the dead. Pico green results demonstrate the same. We looked at the gene targets in the cancer pathways that we expected, cancer-related pathways that we expected to be targets for miRNA-34a 5p, and that was ROCK, TGIF, MYC, and STAT3. We looked at some others also, but we saw, as we had in our microRNA-148b study, substantial downregulation of all of those targets, indicating that we are actually hitting the targets that we think. And again, the mechanism is apoptosis. We then did a bilateral TE10 xenograft. In this case, the mouse got 2 flank tumors. They were grown to approximately a centimeter volume. Then one of those got illuminated. And so you can see in the middle panel there, the red is the unilluminated, the green is the illuminated tumor. The tumor is largely gone again. And then just a graphical representation to the right of the tumor volume. And as you can see, the blue line here is the treatment group. The volume of the control groups continues to increase, and the control groups in this case are vehicle control and the non-light activated. So the scrambled and the non-light activated systems. Both of those, if you don't put any light on it, or if you use a light activated scrambled, the tumor volume continues to increase. When you use the miRNA-34 plus light group, you end up with this massive tumor volume loss, very similar to what we saw in the HRAS squamous carcinoma model. Tumor histology looked very similar to what we had seen before. So in the non-light activated group, we see what we believe to be normal tumor tissue. And in the light activated group, we see this substantial reduction in the light activated plus miRNA-134a 5p, we see this substantial reduction in viable tissue. And so you can see we have normal tissue that changes into this more or less Swiss cheese looking material. So that I'd like to acknowledge all of our collaborators, but then also our funding sources, NSF, the US Army, and NIH. Thank you. Be happy to take any questions.
Thank you, Dr. Hayes. Very interesting work and research. Congrats on all your success and progress there. We have several good questions. We'll start with Dr. Anand Singh out of India, I believe. And Anand asks, why do you see infiltration of immune cells?
So we think in the case of in the first model, which was immune competent, we think it's actually just the degradation of the tissue. So when the tissue starts to die, the immune cells, the immune system responds to that to clear that out as it would any injury. So imagining it was injured tissue. I think the more interesting piece to that is why do we see, even in the non-light activated systems, the nanoparticle itself seems to change the phenotype or the relative amounts of the phenotypes of our T cells, which we thought was an interesting result.
Okay, great. And so I think Anand had suggested here, does this mean that the microRNA 148 induces an immunogenic cell death? Maybe you just answered that.
Yeah, I don't think it directly influences the immune cells. I think it is really that it's driving general apoptosis, and general apoptosis is signaling the immune system to.
Yeah, well, you just described the mechanism, which is what Anand also asked. That was his third follow-up question. And the next question is from Athena Starland Davenport. And Athena asks, could you use your microRNA nanoparticle technology to treat blood disorders?
Oh, possibly. I don't want to say no, but one of the challenges is the light delivery. And so if it's a localized tumor, envisioning delivering the light to that location, particularly if it's superficial, and a lot of obviously what we're doing is head and neck or other superficial tumors, that it's very easily to envision how we would deliver that light. To effectively deliver light to a blood cancer or some other, we may have to take it out, circulate it through a light source. I'm not saying it's impossible. It could be done, but it's a bigger engineering challenge.
You really need a localized focus of the light. It's interesting. It's kind of like the nanoparticles that are activated by radio frequency waves, right? It's work for solid tumors, but for leukemia, for example, it's quite dangerous to put a person through an RF field with metal in their bodies, basically.
Absolutely. And it would be a very similar challenge.
Yeah. Sounds great. And somebody, let's see, a very good question about the exosome. What is the role of the exosome in this system? And this is asked by Babak Bainam.
So we don't know in that, in both model systems, one which had immune cells, one where was immune competent, one was not, we see a really dramatic collapse of the tumor. We don't believe that we're getting 100% uptake by tumor cells or transfection efficiency of the tumor cells in vivo. So is there cell-cell communication after activation, which is driving apoptosis more completely? We're really not sure. I don't have the data in this presentation, but we also don't see these tumors come back, at least not within a couple of weeks, which was the time it took to originally generate them. We really believe we're getting a pretty complete kill of the tumor. So there may be a role of cell-cell communication in there, or exosomes playing some sort of role in that process.
Yeah, there's a question. It's about side effects, and this is from Ankit Suwach. Are there any known side effects to the treatment yet, either through the light administration or I guess directly from the therapeutic microRNA itself?
So the light administration, not really. Certainly we're using an LED. We're not using a laser, so it's not that intense. It's just a regular LED, non-coherent. So the power is not very high. Yeah, it's near IR. You could cook tissue, if you used enough light. So that's certainly a concern. But so far we haven't seen that. We have not been, because this is so efficient in phonon generation, we haven't needed to use that much light. The nanoparticles themselves, the base nanoparticle is metal. The evidence from our studies and others indicates that it does persist in the liver for some time, weeks to months, in reducing amounts. There doesn't seem to be any obvious toxicity associated with that at this point that we know. It's gold and silver. We expose ourselves to gold and silver quite a bit, so it's not the bigger concern would be the particulate nature of it. But again, we haven't seen any obvious toxicity or inflammation of the liver. It's cleared from the other tumors within about 24 to 48 hours. The side effects, certainly if we get leakage in the sense that we are delivering microRNA to non-target tissues, as I said, there's if we look at microRNA-148b, for instance, 6,000 different predicted targets. There's going to be massive side effects. But that is really the key to getting this temporal and spatial control and using an RNA-based therapeutic. It's pretty reliable. Where we shine the light, that's where we get release. And the RNA itself degrades probably within about 24 to 48 hours after administration anyways. So where we don't light activate it, it is probably not going to have a major impact.
Great. And Usman Traore is asking, is it possible to use your base technology here in gene therapy applications? So I guess not microRNA delivery, but maybe some type of long-term gene therapy.
Yeah, so we can deliver bigger constructs. We have not explored it in detail. There's no theoretical reason why you can't put bigger chains on. And we have done some initial studies in that space with some success, but I can't say we have a lot of data on that right now.
Yeah, I think we're getting close to the end. I know you've got a hard stop here in just a few minutes. I'll ask one more question. My apologies to the others who we couldn't get to, but we will make a log of this and get your questions answered individually. Franz Kokaja is asking the question, what's the uptake mechanism of the nanoparticles from the cells? Like, is it endocytosis, receptor mediated, non-receptor mediated? How does it work?
We think it's we don't think it's receptor mediated. We think it's endocytosis and general transport. So I know we think of the membranes as relatively impermeable, but there's evidence that nanoparticles will actually just squeeze through the membrane. So we think there's a combination of endocytotic mechanisms and just uncontrolled transport.
Really exciting technology, Dr. Hayes. We'll stop there because I know you've got another meeting to go to. Very briefly, I just wanted to thank all the attendees for joining us today. We had many, many folks show up from all the way around the world. I'll just name off the countries really quickly because I think it's important. Researchers from Ethiopia, Germany, the Cayman Islands, of course, the USA, Iraq, India, Guinea, Kenya, Turkey, Mexico, the UAE, Singapore, and Iran. So really a world-class group today. We appreciate your timing. We know it's a little bit inconvenient for some of you around the world. I want to thank everybody for joining us. Dr. Hayes, I specifically want to thank you for your lecture today and congratulate you on really all your success throughout your career and specifically this success as well. It's very exciting. And one more thing, I just would like to thank my colleagues, Christine Lay and Lilith Elmore, also for helping to host the webinar today. I think it went off pretty well.
Excellent. Thank you, Rob. And thank you everyone for attending.
Thank you. And everybody have a good evening, morning, or day. We appreciate your time. Thank you. Bye-bye.